So we now know that every section of DNA, called the gene, will code out for one protein. So if you got your DNA, and this section right here is one gene, that means it's going to be the genetic code to make a protein. It's basically the blueprints on how to make some kind of specific thing you need. This is just like our light switch from the other videos. It has to be flipped on by some hormones, by some kind of messenger, by some kind of chemical signal, by some kind of process. But once the light switch is flipped on, I can go through and read the genetic code along that section. So the question is, what do I actually do with that genetic code once I read it? And that is the next process we're going to learn about, which is called protein synthesis. Synthesis basically means to kind of create or make. So if I'm going to go through protein synthesis, it's the process of making a protein. So I have to start at the very beginning. So inside my cell, I have my nucleus. This is where I'm going to keep my DNA. So if the DNA is the blueprints, I need to get that to wherever I make my proteins, which happens to be outside the nucleus. The problem is the DNA cannot leave the nucleus. The DNA needs to be protected, it needs to stay in there. I don't want to risk moving it around the cell and it get damaged, because then I'm going to be reading off broken blueprints. So it will never leave our nucleus. It will always stay there. That means if I need to get that message out of the nucleus, I need to make a copy and then send the copy. I can't send the original. So now I need to break open my DNA and read it. And the first step we need to do is very similar to the same one for DNA replication when I needed to read it and copy that. First off, we need to unzip the DNA. We've done this before, and we're going to use the exact same enzyme that we did for DNA replication. Uncoils and unzips, kind of like unspiraling our double helix like a helicopter. It's going to be a little enzyme called helicase. It's going to travel down, it's going to unwind and uncoil our DNA until we have it opened up so we can actually access the code. Then I'm going to have to read the DNA, and I'm going to have to make my copy. In DNA replication, we read it and made another copy of DNA, so it was called DNA polymerase. I need to make a copy called RNA. DNA is a double strand. RNA is going to be a single strand with the code copied on it that can actually leave the nucleus. So as you might have guessed, the little blob that's going to travel down the side of my DNA and make my RNA copy is called RNA polymerase. RNA polymerase comes in and it's going to read one base at a time along the side of the opened up DNA. So say it's read 3 right there, it's going to spit out a section of RNA with 3. This is going to be kind of like making a photocopy or a Xerox, and then I can send this little strip out, because remember, we are still inside the nucleus. This is going to be still in the nucleus, this is going to be outside. So this little thing right here can actually slip out of the nucleus while the DNA can't. So just like DNA replication also, we have to pair up these bases. Normally we go apples are in trees, cars are in garages. However, this is when we go from DNA side to DNA side. That would be like taking a complementary DNA side and then making the other side apples, trees, cars, garages. RNA, however, does not have one of these letters. It does not have a T. RNA will only have A's, C's, G's, and U's. It's going to have uracil instead of the T. So all the other rules apply. It's just every time I hit an A here, I'm going to make a U there instead of a T. So say I've read those 3 sections, that's the entire gene. The gene to make one protein is only 3 bases. We read it, now I have my strip of RNA. Since this RNA is taking a message and it's sending it out, we refer to this as messenger RNA, or the M is standing for messenger. We've basically taken a message about yourself and sent it off somewhere. So for example, in high school, your senior year, you take a message about yourself, your senior year, and all your grades and everything you've done, and you send it off to colleges, and it's called a transcript. This whole process of going from DNA to messenger RNA is called transcription. And transcription is going to all happen right here inside the nucleus because we can't move our DNA out at all. Little pink helicase is our little triangle, it splits it. RNA polymerase reads and rebuilds a strip of mRNA, and now it can slip out of the nucleus. So we don't know where it goes yet, but I'll tell you what, you're not going to create a protein inside the nucleus. I need to find my protein factory. It just so happens the protein factory is the little dots that we've been drawing in almost every unit called ribosomes. And here I have them drawn as the purple dots. So now we can go to the next step, which is what happens when I actually get my messenger RNA to the ribosomes. So our messenger RNA comes on out. So messenger RNA comes on out in the cytoplasm because that's where the ribosomes are. They're either floating around in cytoplasm or hanging around on the rough endoplasmic reticulum. And now I can start to decode them. So the ribosome actually kind of has two parts. It's got two blobs on it. The messenger RNA is going to run over there, and the ribosome is going to kind of grab it up, and it's going to read each letter. Say, for example, I'd come in with these bases. So each of these bases that I'd copied from the DNA, my ribosome is actually going to read them 3 at a time. This is actually called a codon, sometimes called a triplet. You'll see it written both ways, it kind of depends. A codon or triplet is going to be the 3 bases, so I'm going to read them 3 at a time. That's how I read my genetic code. The letters and the sequence of letters are going to determine what I actually click into place. Remember, we are doing protein synthesis, which means our end goal is to make a protein. We know from biomolecules that monomers are building blocks of a larger polymer, which is the end result, is the biomolecule. If I want to make a carb, I need simple sugars, monosaccharides, all put together until I get a full carbohydrate. If I want lipids, I need fatty acids, glycerol. If I need nucleic acids, I need to put together nucleotides. The monomer of protein is called an amino acid. So if I want to build a protein, I need to click a bunch of amino acids into place, just like Lego blocks, until I get a full polymer, so I can make my meat, my enzyme, my hormone, whatever protein I need for the body. It's built from amino acids. Some of the amino acids you will make in your body, a lot of them you get from your food, but the point is they are all floating around in your body, kind of free floating. The ribosome's job is to grab the correct amino acid that pairs with this right here. Its job is to find the amino acid and click it into place. Think of it as a puzzle piece. You have an amino acid as this part of the puzzle piece. ACG might have its port to kind of click into place. So only a very specific amino acid will go with the very specific letter. So let's assume all these different little colored circles are all different amino acids. You've eaten them or you've synthesized them and made them, whatever. You need to click these into the correct order and place in order to make a full protein, since these are the Lego block monomers. So in order to know where these go, we have to read the code on the messenger RNA. Remember, this is our red strip down here. The ribosome is going to read 3 at a time, and it's going to click something in the place that makes the correct sequence. It needs to find something that bonds with the opposite. Since we are no longer in the nucleus, we still don't have the letter T, we're still using the letter U since we're talking about RNA. There's another type of RNA that we can use here. This is called tRNA. tRNA's entire job is to get an amino acid hooked on top and then find the correct spot to bond it to. Once the amino acid hooks into the tRNA, it floats around and waits, and the ribosome is going to click it into place. So you essentially have all these amino acids lying in wait, waiting for the ribosome to read the code that bonds with them. The ribosome reads the codon and clicks in the appropriate amino acid until they're in the correct sequence. So maybe the sequence is red, yellow, orange, green, blue. At that point, messenger RNA, when it's done building its amino acid, will have something at the end called a stop codon. That basically means stop. It means cut it right there, that's it, that's the whole protein. So as we click in these tRNA, the amino acids are actually going to bond together. Every time we click a new one in, they string together. Because the eventual goal, we don't need the tRNA, we just need to assemble amino acids. So every time it clicks one in, and they connect to each other, they're going to pop off of the tRNA. We no longer need it. The tRNA basically just served to bring the amino acid to where it was supposed to be based on the messenger RNA codon. So they'll start to pop off and form their own little chain. So now we have this. This is a bunch of amino acids, 5 different amino acids all put together. And once they do that, they are going to kind of act like a slinky. A protein will kind of recoil and kind of refold once it's assembled back into its correct shape. The shape of the protein actually determines a lot about the protein. So maybe this is our final assembled protein. Whatever this is, amino acids all formed into this weird looking kind of shape. Then it can go off and do its job. Maybe it's a hormone, maybe it's some kind of actin or myosin, and it can go off and perform its function based on how it's structured. Remember, we learned in cell differentiation that the structure determines the function in cells. For proteins, this is the same way. How they are folded and created and from what amino acids determines what they are going to do. Something easy you'll even recognize is eggs. When you crack an egg into the pan, you have the yolk and the white. And initially the white is clear, like the egg white is kind of clear and goopy looking. As you apply heat, it changes and becomes white and kind of spongy and firm. You have not changed the egg amino acids per se. You haven't changed it into another thing. But what you've done is applied heat so that maybe this right here moved over here. And now we have a very different structure. This is no longer our shape. So we see it differently. It changes the integrity of the protein. And our egg is now cooked. This is why meat will go from red to brown when we apply heat. This is why, if you know what ceviche is, which is where you put fish and shrimp and stuff into lime juice, the lime juice will actually change the proteins and cook them basically by changing how they form. They will change colors. When you apply heat to stuff, it will change the color of the protein. So I cannot just jumble all these amino acids into a ball. They have to be specifically ordered and folded, which is the final form of a protein. So the last step is, how do we figure out which amino acid goes where? So we know that we have a codon here, and it clicks into these 3 at the bottom of the tRNA. So the tRNA is going to have a little spot here, here, and here that matches up with UCA. It's called an anticodon. That makes sense considering it's the opposite of the codon. So if you have U, this tRNA needs to have A. If it has C, it has to have G. If it has A, it has to have U. Remember, it can't have T because we're still in RNA. So that anticodon is what clicks it into place. That specific tRNA with that specific anticodon will have red or yellow or orange or whatever. So we are going to read the messenger RNA to determine what the actual amino acid is. And there's 2 ways that we can do that. So say I give you this DNA strand from the gene. My RNA polymerase ran along and read these bases from the DNA. Remember, this is all still inside the nucleus. ATC, CGG, GCC, TAT, and GCG. So that's what was in my DNA. That codes for something. I don't know what it is yet, but it codes for something. So we gotta follow our rules. We know apples are in trees, cars are in garages for DNA, but we also know that we can't have T in any RNA. So if my next step is to make a messenger RNA out of that gene, I can't use the T, I gotta use the U. That means my messenger RNA based on that DNA gene is gonna look something like this. A goes to U, the A goes to G, C goes to G, goes to C, G goes to C, G goes to C, C goes to G, C goes to G, goes to A goes to U, C goes to A, G goes to C, C goes to G, and G goes to C. So I've made my RNA messenger code. I read the DNA, and then I used my rules to convert it. So that's our step 1. That is called, as you remember, transcription. So now I got messenger RNA. Now I have to figure out that second step. Right now the RNA is gibberish to us. Think of it as another language. I need to take that and put it into a language I understand. When you take a language you don't understand and convert it into something that you can read, that's called translating. It's called translation. So it makes sense that protein synthesis is translation. This is when I take my messenger RNA string, I click in my anticodons, which are at the end of my tRNA, and at the top of my tRNA I have an amino acid bonded to it. So now every time I click in a new amino acid with the ribosome, those amino acids are going to bond into a string, into a polymer called amino acid chain or polypeptide chain. It's the same thing. So we have to figure out how to do this translation process. I have to figure out which colored amino acid circles I click in where. So here's my wheel. Each of the letters on the outside, the 3 letters, like THR, ASN, LYS, stand for one amino acid. That is one of the colored circles. We can actually figure out which colored circle we're going to get based on what the RNA is. So let's look at our first RNA codon. And we're going to go through the translation process with UAG. For the wheel, we start here in the center. I'm going to go to U. That means that within the U pi piece, I'm going to look for an A. Here it is right here at that green part. Now within the A pi piece, I'm going to look for a G. So UAG actually codes for stop. Maybe that was the end of an RNA protein string, and now we can start up the next one. So let's do the next one, GCC. Start in the middle, G. Within the G pi piece, I find my C. C. Within the C pi piece, I find the C, which gives me ALA as my amino acid, alanine. Then I go to CGG. We do the exact same thing. C, G, G, ARG, arginine. Some wheels will have the full thing written out. Some will just have the 3-letter abbreviation. It's not a big deal. Doesn't matter. AUA, A, U, A, ILE. Put our isoleucine right here. And then finally CGC. Start in the center. C, G, C. ARG. Look at that. And that is our entire amino acid chain, which would form the polypeptide chain, which would then form into the protein. And that protein can go off to be the phenotype of one of our traits. It can be some specific little structure we need. It can be any of the protein functions. Bear in mind, we just made up some random letters for DNA just to practice. In an actual amino acid chain, you are always going to have a start codon over here on the wheel. See how MET, which is AUG, codes for start. That will basically tell your ribosome that that is the start of a new sequence. So methionine will kind of be like, okay, it's always going to start with methionine. You also have stop codons. We saw one pop up in our example by chance, but UGA, UAA, and UAG are all stop codons. That is going to cut the end of that amino acid chain, so that's basically it. And it'll form up to its protein. It's going to make sure you don't get these long, crazy runs of proteins, which can cause a lot of problems because you don't want a protein with 20 amino acids when it only needed to have 4. That is the amino acid wheel. There is also something called a codon chart. This is how I learned it in high school. The wheel I kind of like better, but it doesn't make that much of a difference. Like, neither one is so complicated that you can't do it. Plus, you're kind of responsible for knowing how to use both. Some questions will only give you the chart, some will give you the wheel. I mean, technically, I guess you're free to flip through the test and find a question that gave you the other one, but this isn't really that hard. So I'll show you the exact same thing with our codon chart. So we'll start with our ATC to UAG again. So we got to find UAG. We'll do green as the first base, red as the second base, and blue as the third, just so it's easy to kind of follow. So I'm going to look on the left. This is going to be my first base. UCAG. I gotta choose my U, so I know right off the bat then my amino acid is going to be in this first row. Then we go to the second base. It's an A. Second base is listed up here. So I know it's going to be in this column because that's where the A is. I basically battleship these up very similar to Punnett squares, so I now can see where they intersect. My amino acid has to be in that box. Third base is these letters all the way over here. We are going to look basically in which row of that box G appears. In this case, G appears right there. So now I can automatically tell that's going to be our stop codon. So the end amino acid is where the left first base, the top second base, and the right third base all intersect. In this case, it intersects right at our stop codon. And as we look over here, sure enough, that's what we got. So the codon chart is not super complicated. It's a little bit different. Some people think it takes longer. I think the wheel is a little bit smoother, but it doesn't necessarily matter. And that basically brings us to the end protein. So we've completed the entire process. We've taken transcription. We've gone from DNA to messenger RNA. The messenger RNA leaves the nucleus because the DNA can't get out, and it floats into the cytoplasm towards our ribosomes. So once we get to the ribosome, it'll start to read our mRNA codons 3 at a time. It'll click in a tRNA with the correct anticodon, just like a puzzle piece. That tRNA is going to have our amino acid on the top. It could be whatever amino acid our Wheeler chart says, and it's going to connect those amino acids together in the sequence they're put in. So we've basically gone from mRNA to amino acids in translation. The amino acids, or the polypeptide chain, forms up in the proteins, and there you go. There's your actin for your muscle, there's your myosin, there's your hormones, there's some kind of structure, and it's all based off the original blueprints from the DNA.